Good afternoon, folks. It's great to be here and great to see all of you. How many of you were very excited to be reminded about how old you were this morning, first thing? When you look on our website, we definitely average out to boomer. So that says something about us, but we've also got somebody who wasn't on the chart. Our chairman is kind of a neo greatest generation guy. So we're pretty proud of Bob, and he couldn't make it this time because he just had eye surgery, but thrilled that we're a part of this and great to be associated with OIS and Aura. I want to talk about visacomatide, which is also ST100, which is our comprehensive therapeutic for dry eye disease. We're a late clinical stage company. The peptide that we're developing is a synthesized polypeptide that mimics a fractional strand of human collagen. The way it works is it binds to disease-damaged collagen. That binding provides a number of features that we'll talk about here in just a few minutes. And we had a pretty interesting journey as we went to our phase 2 trial and then digging even deeper into the mechanism of action I'll share with you today. Collagen is a biologically active and very important component of the eye and the optic nerve system in the human body. It is a cell signal ligand binding site for important proteins. It helps the microenvironment stay healthy in homeostasis. When we repair damaged collagen, we help restore that homeostatic role for collagen. And collagen damage is pathogenic in a number of ophthalmic indications, including dry eye, Sjogren's, neurotrophic keratitis, et cetera. The first experiment that we ran, the first study we ran with the collagen mimetic peptides, or CMPs, was a lamellar keratectomy, where we removed the epithelial layer and some of the stroma edge to edge on mouse eyes. And we put a very low concentration single drop of the peptide on these animals just to see what would happen. We had really no idea. This was our first foray into this. And what we found was that, sure enough, in 16 hours, we had full recovery of the corneal epithelium, full adherence to a basement membrane and the stroma, and significant reduction in inflammation signs in the stromal tissues. So, hey, something that deals with one of those things we talked about earlier today, which is corneal ocular surface tissue health. Very good thing. We thought we got there. So let's go crazy and let's do a phase 2 clinical trial, which we did. So the ST100 phase 2 trial addressed visual function, symptoms, staining, and lacrimal tear production within 28 days. We hit a visual acuity endpoint with significance on the second and third day. We had symptom improvement by the second week, staining by the week 4, and lacrimal tear function by week 4 as well. The drug is very well tolerated. The safety profile is very nice. The drop comfort scores were similar to typical artificial tear products that patients are using today, like Sustain. When we looked at all those endpoints and the way that all came together in that trial, we said to ourselves, okay, we think what we're having here is an impact on the lacrimal function overall. So let's take a look at a couple of different animal studies that'll help document that and give us a feel for exactly what else is going on with this peptide in the eye. So we did a study using an atropine and a desiccation chamber to create ocular surface damage in mice and corneal nerve damage. And as you can see in the images across the top, which is the naive animal, it's got an intact subbasal nerve plexus swirl pattern. You've got the corneal epithelial terminals in the next image, and then a zoom in on those in the third. With the vehicle animal, you got obliteration of that subbasal nerve plexus and massive reduction in the epithelial terminals. Conversely, when we took those animals that had been subjected to that same damage and treated them with ST100, we got a recovery of the subbasal nerve plexus and a restoration to near naive levels on the epithelial terminals. So nerve damage, nerve recovery, another key piece of dry eye disease was happening with this peptide. And that got us very excited. Anatomical impact on an important factor in the pathophysiology. We also looked in those same animals at the lacrimal reflex synapse in the trigeminal V1 structure. We examined those tissues, and as you can see on the right-hand side of the image, The vehicle animal has a significant dropout in cell body area in V1, which is the part of the switch process for the tear ducts. Conversely, the CMP caused a significant amount of recovery in the cell body area in the trigeminal V1. We also measured something called VGLUT2 expression, which I didn't know about until we did this study. But in any event, it kicks off and elevates when you've got significant nociceptive pain activity on the ocular surface, or if there's significant corneal nerve damage. So this provided us with an explanation for the impact on the lacrimal functional unit. Now, this data is brand new, is about a week ago, and we did a similar type of study, and we looked at mast cells, and we wanted to look at where we had degranulation of the mast cells in the lids of mice and compare that with treatment. And as you can see on the left-hand side of the image, the brighter, bigger dots are the degranulated mast cells, which are releasing histamines and inflammatory cytokines, whereas on the right-hand side of the image, the treated animals, those number of those degranulated mast cells or activated mast cells is vastly reduced. So this gives us another piece of data about the innate immune system and the innate immune response in animals and the impact that we're having on that. We've done prior studies on inflammatory impact where we looked at the expression of inflammatory cytokines on damaged collagen substrates by epithelial cells and then treated to repair those substrates and saw a return back to naive levels on IL-6 and IL-8 in this particular study. And it's well established in the literature that healthy collagen is a ligand binding site for LAIR1, which is expressed by a number of circulating inflammatory cells. Healthy collagen binds to LAIR1, prevents the activation of those circulating inflammatory cells. LAIR1 will not bind to damaged collagen, so our repair of helical collagen structures in the eye, in particular as it relates to dry eye disease, provides us with an improvement in the expression of circulating inflammatory cells. When you combine that nerve repair, that multimode anti-inflammatory effect, et cetera, it gives us a pretty good explanation for what we saw in the controlled adverse environment chamber during our phase 2 trial. Patients on successive chamber visits at day 7, 14, and 28, these are area under the curves of their discomfort scores, and going down the chart is an improvement, by the way, saw successive improvement in their overall discomfort relative to vehicle every time they came into the chamber. So increased consistent use of the product, saw beneficial effect to the patients in terms of symptoms. The protective effect grew with regular use, and we think that this gives us an opportunity to prevent recurrence of symptoms with the drug if it's regularly used. We are running our phase 3 clinical trial right now here in the US and in Australia. We've got 350 patients we're going to run through the trial, and we're very excited about the opportunity to be here today and talk to you a little bit about the drug. We think that we hit not only the nerve damage question in dry eye disease, which is important as we saw in DUCE 2, but also symptoms, issues, ocular surface tissue recovery and repair, and a broad anti-inflammatory impact on patients that suffer from dry eye disease. I really appreciate the opportunity to talk to you folks today, and if you've got questions, look me up. Thank you so much.